One of the most important archaeological discoveries of the past century, if not of all time, occurred in 1947. Our understanding of the Bible completely changed, and it happened because of a sheep.
Imagine uncovering ancient manuscripts that have shaped biblical scholarship for decades, and then discovering that the very material they're written on tells a different story altogether. By sequencing DNA from the parchment of the Dead Sea Scrolls, scientists have upended long-held beliefs about who made these scrolls and where they came from. What if these scrolls weren't the exclusive collection of a single isolated sect as once thought? What if they represent a complex tapestry of communities and ideas hidden for 2,000 years in the genetic code of animal skins? The puzzle of the Dead Sea Scrolls. The Dead Sea Scrolls are one of the most significant archaeological discoveries of the 20th century, yet they remain a mystery wrapped in thousands of tiny, fragile fragments. Scholars have been painstakingly piecing them together for over 70 years, but the sheer volume and condition of the scrolls have made reconstruction incredibly difficult. With about 25,000 pieces representing roughly 1,000 different manuscripts, many fragments are just a few words or partial lines on delicate parchment, without titles, page numbers, or clear markers. Determining which pieces belong where has been like assembling 1,000 incomplete jigsaw puzzles without a guiding picture. Traditional methods relied heavily on handwriting analysis, comparing scripts to detect if different fragments were written by the same scribe. Researchers also examined physical clues, texture, color, thickness, trying to match pieces that might have come from the same animal skin. Textual links were sought by analyzing content for connections or thematic similarities between fragments. Despite these exhaustive efforts, the process was slow, uncertain, and fraught with risk. A wrong match could create false interpretations, attributing ideas to ancient authors that never existed or merging texts that never belonged together. The potential for error threatened to distort not just individual works, but decades of scholarly understanding. On top of these complications, the scrolls' extraordinary fragility has only increased over time. They have suffered from natural aging and exposure despite the dry climate that preserved them initially. Many fragments are now too damaged or tiny to be confidently read, and because they are sacred texts for multiple religious traditions, any destructive testing is strictly forbidden. This meant scholars were limited in the tools they could deploy, forced to rely almost entirely on non-invasive traditional techniques that could not answer some key questions. On a deeper level, the scrolls posed a profound intellectual challenge. They do not present a uniform message or single origin story. Instead, they include a bewildering diversity of scripts, scribes, and styles. Some fragments showed conflicting versions of biblical books, different writing conventions, and vast variation in parchment quality. This inconsistency puzzled experts, offering hints that the scrolls might come from far more complex origins than previously assumed, but leaving the door wide open for speculation. Despite all this, the dominant view took shape over decades. Scholars reached a consensus based on the best evidence and interpretation available. This rigid framework shaped how the scrolls were understood in academia and beyond, influencing perceptions of early Judaism and even Christianity. Yet behind this consensus, nagging questions persisted, questions traditional methods couldn't fully untangle or resolve. The mystery of the scrolls' origins lingered like a shadow, growing as more fragments were studied without definitive answers. The puzzle was enormous. Who wrote these texts? Where did they come from? And why do they present such diverse traditions if they belonged to one group? These were not minor academic details. The answers held the power to reshape our understanding of religious history in a critical period. For decades, scholars were trapped by the limitations of their tools and the sacred nature of the texts themselves. They were driven by the hope that new evidence would one day emerge to illuminate this ancient enigma. What they didn't expect was that the breakthrough would come from an unlikely field, genetics, offering a microscopic key to unlock millennia-old secrets. The theory that best explained the scrolls for so long now requires fresh scrutiny, especially given the insights about their origins and provenance. The Essene hypothesis. For much of the past century, scholars have accepted the idea that the Dead Sea Scrolls were the private library of the Essenes, a Jewish sect described in antiquity by historians like Josephus and Pliny the Elder. According to this theory, the Essenes lived in a monastic, isolated community at Qumran near the Dead Sea. They followed strict purity laws, maintained a rigorous religious lifestyle, and copied sacred texts, preparing for an apocalyptic future. When the Roman army advanced in 70 AD during the Jewish revolt, it was believed the Essenes hid their collection in nearby caves, where it would remain buried for nearly 2,000 years. This theory neatly explained many apparent features of the scrolls. The sectarian writings, emphasizing communal rules, aligned with historical descriptions of Essene life. Calendrical texts showed a solar calendar distinct from the lunar calendar officially observed in Jerusalem, supporting the existence of a separatist religious community. Apocalyptic writings expressed hostility toward the Jerusalem priesthood, fitting the image of a radical group awaiting divine intervention and rejecting mainstream Judaism. For decades, this interpretation became the scholarly and public standard, taught extensively in universities and shared widely in documentaries and exhibitions. The Essene hypothesis provided a convenient and coherent narrative, bridging archaeological discoveries with ancient historical accounts. It allowed scholars to frame the scrolls as the uncompromised religious voice of a single, tightly knit group, placing Qumran at the center of this spiritual world. The idea was compelling and persuasive, dominating biblical and archaeological scholarship for generations. Yet, experts faced ongoing difficulties about the theoretical framework. The scrolls themselves displayed major internal inconsistencies which did not match the expected pattern of a complete sectarian collection. The manuscripts displayed multiple textual representations which different scribes and writers used to create their content. The radiocarbon dating established that some scrolls existed before Qumran became a settlement, which created problems for dating the library of one particular community. The parchment exhibited different levels of quality, which included both expertly treated skins and poorly handled materials that did not match the standards of a properly maintained monastic scriptorium. Most puzzling of all, the theological diversity within the scrolls seemed at odds with the idea of a strictly uniform group. Some texts contradicted one another or preserved traditions that the Essenes would likely have rejected. If the collection belonged to an isolated sect, why would it contain such conflicting versions of biblical books or wide-ranging religious perspectives? The different elements within the system made it more complex than the prevailing theory which considered it to be a unified organization. The Essene hypothesis remained the most viable explanation because researchers needed additional evidence to support their claims. The theory obtained deep acceptance, which created a framework for understanding, yet left major issues to be resolved. Scholars continued to grapple with these contradictions, searching for new methods and data that might illuminate the scroll's true origins. Extracting and interpreting ancient DNA from the scroll's parchments revealed truths hidden beneath the surface of tradition and script. DNA comes into play. The idea that parchment, made from animal skin, might contain retrievable ancient DNA was a game-changer. It started when molecular biologist Oded Rechavi and biblical scholar Noam Mizrahi realized that every fragment of parchment carried biological information locked inside its collagen proteins. This DNA could offer insights into the origin of the scrolls without damaging the sacred manuscripts. But how could they extract usable genetic material without cutting into the fragile, priceless texts? The solution came from decades of handling and storage. Minute particles of parchment dust had collected in the containers where the scroll fragments were kept. This dust, from damaged or margin areas free of writing, contained enough collagen to isolate ancient DNA safely and non-invasively. Scientists could now analyze these tiny samples, circumventing the ethical and conservation restrictions that forbade direct sampling of the texts. It was an ingenious way to harness what was previously considered waste to unlock new information. The process of extracting DNA from ancient parchment proved to be extremely difficult. because of its unique scientific requirements. The parchment-making process destroyed DNA through molecular damage, while the scrolls used iron-based inks, which further destroyed their genetic material. The remaining DNA, after 2,000 years of desert environmental changes and 70 years of human contact, showed severe DNA degradation, together with modern human DNA contamination from multiple researchers who handled the samples. The researchers established strict ancient DNA laboratory protocols, which required complete protective equipment and used sterilized work areas while operating positive airflow systems and conducting ultraviolet light decontamination treatments. The researchers selected mitochondrial DNA for their study because its presence in each cell as multiple mitochondria boosts their chances of obtaining useful DNA fragments which become damaged over time. Nuclear DNA identification proved challenging, yet it revealed vital details about specific animals and their genetic connections to other animals used in parchment production. The research team used multiple sample extractions together with interlaboratory testing and statistical analysis techniques to confirm their authentic ancient DNA results while separating genuine ancient DNA from modern contaminants. The project required thousands of hours and hundreds of samples for completion, yet it achieved success through the acquisition of genetic material from numerous scroll fragments, which marked an unprecedented accomplishment in scientific history. The discovery provided scholars with a scientific method which uses objective evidence to connect scroll fragments and establish their historical roots. Because the method exceeds traditional methods of handwriting identification and parchment examination. The research opened new research possibilities to study the composition and provenance of scrolls together with the historical communities who created them. By reading the DNA hidden in the very material of the scrolls, we begin to glimpse the untold history encoded in the genetic makeup of their ancient animal sources. Genetic revelations. When researchers began sequencing ancient DNA from the Dead Sea Scrolls, the first surprise was the remarkable variety of animal skins used to make the parchments. Most fragments came from sheepskin, which makes sense given the animal's prevalence in ancient Judea and its adaptability to the harsh desert environment. Sheep were the natural choice for parchment due to their abundance and the relative ease of raising them in arid conditions. But then came the unexpected discovery. Several fragments were made from cowskin. This detail flipped the established narrative on its head. Cattle are not desert animals. They require plentiful water and lush grazing land, conditions absent from the Judean desert around Qumran. The discovery of cow parchment fragments implied that some scrolls had to have originated far from Qumran, in regions with more abundant resources capable of supporting cattle herding. This meant the scrolls were not simply the output of one isolated community, as previously believed. Instead, they represented a far wider geographical and cultural constellation. The implications broadened further when geneticists analyzed relationships between animal skins. By examining mitochondrial and nuclear DNA, they developed fingerprints that could link fragments to the same individual animal or to animals closely related to one another. This scientific approach gave scholars a revolutionary, objective way to group fragments together, surpassing the subjective and often tenuous judgments based on handwriting or parchment appearance. The case showed different versions of the biblical book of Jeremiah through its presentation. The DNA evidence demonstrated that two different textual traditions existed because the longer version used sheep parchment, while the shorter version used cow parchment. The two versions proved their origin from different communities who lived in different geographical areas. The traditions transmitted through their independent preservation maintained actual differences without any copying errors or scribal differences. The analysis, which examined songs of the Sabbath sacrifice found at Qumran and Masada, discovered multiple copies, which were made from animal skins that did not share any common characteristics. The text demonstrated its widespread popularity because it reached different communities, which undermined the belief that such writings exclusively belonged to one sect. The genetic variation showed strong manuscript production networks which operated throughout the region of Judea. The researchers discovered kinship markers through their research of parchment materials, which created new understanding about parchment origins.
You can observe various animal parts, and that's the best way to do it by your hand.
The research team found animal skin clusters that showed genetic similarities between siblings, parents, and offspring, which indicated that manuscripts had been created at local centers which used herds to produce multiple scrolls. The collection consisted of two types of items, which included local products and imported goods that came from faraway locations. Far beyond the biology of animals, these discoveries questioned foundational assumptions about the scroll's origins and significance. Rethinking origins and diversity. The DNA evidence dealt a serious blow to the long-held Essene hypothesis, which posited that the scrolls were the isolated library of one sect living near Qumran. If the scrolls came from different animals raised in distinct environments, then the idea of a single community producing and preserving this collection became untenable. Instead, the scrolls represent a curated archive comprising multiple communities, traditions, and geographic regions. This shift reframes the scrolls not as a closed theological statement, but as a snapshot of the vibrant and diverse religious landscape of Second Temple Judaism. The coexistence of multiple versions of biblical books, conflicting ritual laws, and varying scribal methods makes sense in this broader context of diversity rather than in a narrowly sectarian one. It reflects the reality of competing and evolving religious ideas circulating in close proximity before the destruction of Jerusalem. The cow parchment fragments specifically demonstrate that texts physically traveled between wetter, fertile areas and the arid Judean desert, revealing networks of communication, exchange, and possibly pilgrimage across Jewish communities. This complicates the notion of Qumran as the scroll's exclusive locus, suggesting instead that the caves functioned as a geniza, a sacred storage space where worn or problematic texts from multiple groups were gathered for safekeeping. This geniza interpretation also explains why some scrolls predate the Qumran settlement, and why the collection includes theological viewpoints that would have clashed under a single sect's control. Rather than inventorying a unified ideology, the scrolls preserve overlapping and sometimes contradictory voices, encapsulating the religious ferment of their era. This reinterpretation carries profound implications for biblical studies and the history of Judaism. It reveals that the biblical text was not a fixed, uniform scripture during the Second Temple period. Instead, diverse textual traditions competed, coexisted, and circulated widely, complicating the process of canonization. This fluidity and contested authority shaped the religious environment from which both Rabbinic Judaism and Christianity emerged. By exposing the scrolls as a mosaic of communities rather than a homogeneous sect, the DNA findings enrich our understanding of the cultural and theological dynamics that influenced early Jewish thought and the nascent Christian movement. The diverse debates over law, messianism, purity, and authority recorded in the scrolls gain new historical context, grounded in geographic and material reality. With this new scientific lens, scholars can pursue questions that were previously unanswerable. Can they identify distinct production centers? Can textual traditions be mapped to regions? How did these texts move through social and religious networks? These insights urge us to reconsider long-standing narratives and embrace the complexity of history, where every fragment carries multiple layers of meaning: textual, cultural, and biological. Broader impact on biblical scholarship. The DNA analysis of the Dead Sea Scrolls has transformed more than just archaeology. It's reshaping our entire understanding of ancient Judaism and the roots of Christianity. By proving the scrolls were not the product of a single isolated sect, but a diverse collection from multiple communities. The findings force historians and biblical scholars to reconsider how sacred texts were developed and transmitted. This diversity shows that religious traditions were much more fluid and contested than previously thought during the Second Temple period. Before this breakthrough, many believed there was a definitive version of biblical texts that communities universally accepted. The reality, as now evidenced, was quite different. Multiple textual traditions coexisted, some even conflicting, reflecting a vibrant religious dialogue taking place across Judea. This complexity explains why the process of canonization, deciding which texts were authoritative, was gradual, fraught, and ongoing during this dynamic period. Understanding this rich theological ferment gives critical context to the environment in which both Rabbinic Judaism and early Christianity emerged. The scrolls reveal debates over scripture, law, messianic expectations, and purity regulations that shaped the spiritual landscape of the era. This religious plurality echoes into the New Testament as well, helping explain the diversity of early Christian thought and practice in its Jewish surroundings. The genetic discoveries enable researchers to track how texts moved between different communities. Scholars can now use material evidence together with textual analysis and genetic data to create maps of ancient religious communication networks and manuscript production routes. The research combines multiple academic disciplines, which creates a better understanding of ancient cultural exchange than any single method. The findings challenge Qumran's established historical function, which scientists have accepted for many years. The site becomes a central point which connects different aspects of Jewish religious life because it retains texts from multiple cultural backgrounds. The scrolls become a collective voice which presents multiple beliefs instead of representing one unified belief system. The religious beliefs of contemporary faiths now depend on this fundamental shift, which creates new relationships with their original sacred texts. Scripture exists as a historical document which developed through different paths of discussion and historical transformation. People need to understand this complex situation because it helps them understand the stories that exist behind these sacred texts, which will then lead them to discover the different ways faith has developed throughout history. The combination of DNA research with advanced scientific methods, such as multispectral imaging and ink analysis, will lead to new scientific discoveries as researchers continue their studies. The researchers are developing genetic databases from both ancient parchments and animal remains to achieve better origins tracking of specific scrolls. The DNA sequencing of the Dead Sea Scrolls has rewritten the history of these ancient texts, revealing a vibrant, diverse religious world rather than a single sect. This breakthrough shows how science can unveil stories hidden deep within history, transforming our understanding of faith and culture. If this discovery fascinated you, please like the video and subscribe for more stories where cutting-edge science meets timeless mysteries.